Good day to everyone joining us, and welcome to today's X-Talks webinar. Today's talk is entitled In Vitro Safety Screening for Clinical Liabilities. My name is Andrew Jernan, and I'll be your X-Talks host for today. Today's webinar will run for approximately 60 minutes, and this presentation includes a Q&A session with our speaker. This webinar is designed to be interactive, so please feel free to submit questions and comments for our speaker throughout the presentation using the questions chat box, and we'll try to attend to your questions during the Q&A session. This chat box is located in the control panel on the right-hand side of your screen. If you require any assistance, please contact me at any time by sending a message using this chat panel. And at this time, all participants are in a listen-only mode, and the presentation slides will advance automatically for you. Please note that this event will be recorded and made available to you for future download. And at this point, I would like to thank Eurofins Panlabs, who have helped develop the content for this presentation. Eurofins Panlabs is a global CRO specializing in discovery pharmacology testing services. They've been in continuous operation for over 40 years, serving the pharmaceutical and biotechnology community, setting benchmarks for quality, convenience, and scientific expertise. Their mission is to provide pharmacological testing services that predict clinical effects. Eurofins Panlabs' scientific portfolio consists of over 1,350 tests, ranging from molecular assays to cell-based models through proof of concept in vivo activity determinations. They are an extension of their clients' capabilities, providing unrivaled pharmacological expertise and knowledge, superior data reliability, and innovative solutions for drug discovery. Now, I would like to introduce our speaker for today's event, and that is Gonzalo Castillo, PhD, Technical Director, Eurofins Panlabs. Dr. Castillo holds an undergraduate degree in biochemistry from Universidad de Concepción, Chile, and a doctorate degree in molecular pharmacology from the Albert Einstein College of Medicine. His postdoctoral work was carried out at the Dana-Farber Cancer Institute, Harvard Medical School. And without further ado, I'd like to hand the mic over to Dr. Castillo. Dr. Castillo, you can begin your presentation when ready.
Thank you, Andrew. And thank you very much for giving me the opportunity to talk to you today about the safety screening for clinical liabilities. So what I'm going to do today, I'm going to split the presentation in 5 different areas. The first one is going to be, I'm going to talk just for a couple of slides about what do we do, and then I'm going to move into what is in vitro profiling, because that is the major activity that we do in our services, and why it's so important to do in vitro profiling and how to do it. And I'm going to finish with ways to analyze pharmacological profiling using tools like bioinformatics. So who we are. So Eurofins Panlabs has 2 sites in the world. One is located in Bothell, Washington, and the other one is located in Taiwan. Our site in Bothell, Washington is our Center of Excellence for Cellular Services, and their focus is in oncology, immunology, and cellular toxicities like cardiotoxicity, hepatotoxicity, and genetic toxicity. The site in Taipei, Taiwan is our Center of Excellence for Molecular Pharmacology, and the focus of the site is safety, is de-risking compounds. And we do that by testing enzyme, radioligand binding, functional pharmacology, cellular and tissue assays. We also have microbial assays and in vivo capabilities like non-GLP safety assessment. But like what I mentioned before, the main focus is going to be in de-risking compounds. Like what Andrew mentioned, we've been in business for over 40 years, and we currently have over 1,500 testing protocols. Now, we are very unique in the CRO industry because we go all the way from molecular to animal, and we test about 50,000 compounds per year, doing this for over 600 clients annually. The primary focus, as I mentioned before, is the de-risking compounds, and to do that, we have almost 850 molecular assays that include GPCR receptors, ion channels, enzymes. We also have functional assays. We have over 500 of them. And what's really important is that we will have a functional assay that will help to determine if any of these molecular assays is active or not. We also have in vivo assays. We have 254 total. Now, always people ask us, why do you have so many assays? And the reason why we do that is simply because we want to provide you the level of confidence that you need when it's time to make decisions regarding your compounds. So what is in vitro profiling? In vitro profiling is the screening of compounds in a broad panel of targets to identify activities not related to the therapeutic target. Those are going to be off-target activities, and in many cases, they're going to be clinical liabilities. Panels include enzymes, receptors, ion channels, and transporters. An example of a typical in vitro pharmacological profile is shown here on this side of the screen. You probably cannot read it, but what it is large panel of targets tested at one concentration, and there you can see the result. And on the right side is just a graphical representation of the data. And I will be showing throughout the presentation many examples of this type of profiling. But why is so important to profile? The answer actually is shown in this slide. And what you see here, you see the trends of attrition rates of drug in drug development projects. In the y-axis, you have the attrition rate of compounds, and in the x-axis, you have the year from 1919 all the way to 2004. And what you can see, and what is so striking about this slide, is that there is an increase in the attrition rate of every single stage of drug discovery process. You can see a 10% increase from 1990 all the way to 2004 in preclinical, but you see an even larger increase in phase 1. This is a 15% increase. The numbers get bigger as you move along to phase 2, and you can see that there is a 25% increase in attrition rate, and a staggering 30% for the increase in attrition rate in phase 3. So why is that? Why do we see such an increase in the attrition rate of compounds in all these stages of development? And unfortunately, there is not one single answer. There's many different answers. Part of it has to do with the fact that we are targeting more complex targets. Part of it has to do also with the fact that the regulatory agencies are getting more, they're scrutinizing more the submission of compounds. So it's a very difficult problem to tackle. But the question is there any success story that we can capitalize on, that we can build on? The answer is yes. There is one success story, and this is shown in this slide. And what you have in this slide is the attrition rate for compounds in clinical safety, efficacy, formulation, ADME PK, commercial toxicology, and cost of goods. And you can see that there is in two areas there is a decrease in the attrition rate. One is there is 3% decrease in attrition rate in efficacy, but there is a 30% decrease in attrition rate for ADME PK. So this is a very successful program, and it goes all across the industry. And why is this? Why the industry was able to decrease the attrition rate of compounds in this particular area? The reason is because there was a tremendous amount of effort, there's a tremendous amount of investment and an effort from the pharmaceutical industry to move ADME PK early on in the drug discovery process. And the payoff is enormous, because the later in the development that the compound fails, the more expensive that it is for any clinical program. So can we do that in molecular profiling? And that is what we are trying to do. And there is many advantages for in vitro pharmacological profiling. The more important one is that it allows for the very early detection of off-target activities and clinical liabilities. And the same information that you obtain from a clinical profile can be used for structural activity relationship studies. Now, another advantage is that most of the panels are composed of safety targets. And one of the more important ones is shown here, is that the species that they're using, the targets that we're using for safety panels, in most cases are human. in vitro profiling is very cost effective, and since it's so cost effective, then we can actually test compounds, not only the compounds that are going to be moving in the pathway, but can also be testing metabolites. And the reason why this is so important is because often the adverse reactions are caused by the metabolites of these drugs. Now, follow-on assays can be done very quickly. So if you have a hit, you can also determine very quickly if you have agonism or antagonism. And here comes a really big advantage of in vitro pharmacological profiling, is that you can determine agonism and antagonism in not only the target that you're testing, but also in other species, including, in many cases, the species in which you will be doing efficacy studies. The turnaround is very fast. Normally, studies can be done within 5 to 3 weeks, and allows for the side-by-side comparison between the test compound and the lead compound in one particular therapeutic area. Basically, you can benchmark. And another big advantage of molecular pharmacological profiling is that allow you to use tools like bioinformatics, and then you can get insight into activities related to your compound. These are some examples of some of our panels, but there's many panels in the industry, and many CROs have their own panels. But this is what we have, and I'm just going to show you 4 of our panels. We have many panels, but these are the more used ones. And what you have here is, the first one is a hit-to-lead optimization panel. This is called hit profiling screen. It's a very small panel, 30 targets, and the reason why the advantage that we have with this panel is that it's very fast. So the data can be returned to the clients very quickly, within 5 days. Lead profiling screen is our most popular panel, and is used for lead optimization, and is composed of 68 targets. Once you are close to an IND submission, then the panel of choice is going to be spectrum screening. And this is a 168 targets, and the reason why it's so broad is that once you're close into an IND submission, you're going to be pan screening. You want to gather as much information as you can in order to submit to have a successful submission to the IND. But the panel that I'm going to be focusing on today is our new panel called SafetyScreen. And the reason for this is that it's a very unique panel. It's composed of enzyme targets and binding targets, ion channels. And the purpose of this panel is one and one only, and it is detect clinical liabilities. So the idea of this panel actually came from a paper that was published by 4 leading pharmaceutical companies, AstraZeneca, GlaxoSmith, Novartis, and Pfizer. And the paper was published in December of 2012. And these 4 groups from these big pharma companies, actually, they got together. And this is for the first time ever, these 4 groups, they decided to share the rationale, strategies, and methodologies for in vitro pharmacological profiling. But not only that, they also came up with a panel, and they came up with a rationale for this panel of targets, and the focus of this target was, of every one of these targets, was exactly what our SafetyScreen panel is: detect clinical liabilities. The panel SafetyScreen is shown in this slide, and what you can see is that there is 87 targets. There is a combination of targets that include ion channels, GPCR, non-GPCR receptors. You have enzyme assays, and the common theme among all of them is that they're all relevant safety targets. SafetyScreen includes every single target that was recommended by the pharma recommendation. Now, what type of adverse events can we see, can we detect utilizing this SafetyScreen panel? That is shown here, and this is just the adverse event for a very few number of targets. But you can see, for example, for acetylcholinesterase, the main adverse event is going to be skeletal muscular paralysis. Also, you're going to have cardiovascular collapse. For sodium potassium ATPase, you're going to see cardiac arrhythmia and atrial fibrillation. For COX-1, the adverse event is going to be cardiotoxicity. You're going to have bleeding in the stomach, ulcer. For phosphodiester APV4, you're going to have gastric stasis and vomiting and emesis, and on and on. So every single one of these targets, here, for example, ventricular arrhythmia, serotonin 5-HT2B, you're going to have, again, cardiotoxicity. So every single one of the 87 targets is going to be associated with a severe A-type of adverse reaction, enough to derail a program from the drug discovery pathway. Now, the adverse events that on SafetyScreen, they're going to fall within one of these systems. So for cardiovascular, the type of adverse event that you're going to see are going to be blood pressure, cardiac inotropy, arrhythmia. For GI, for gastrointestinal, you're going to see compounds that will be affecting GI motility, emesis, gastric emptying. For CNS, of course, you're going to have very severe ones that are going to be cognitive functions, sedation, dizziness. Pulmonary, you're going to have pulmonary bleeding, hypertension, bronchodilation. For the immune system, of course, the main one is going to be immunosuppression. For renal, very severe ones are renal dysfunctions, because renal kidney does not have much tolerance for renal dysfunction. And in development, the main ones are going to be cardiac defect during development. So you can see that all of these adverse events will be enough, as I mentioned before, to derail a program from the drug discovery pathway. But if you have a K9, you're going to see that we're missing one, very important one, and it is hepatotoxicity. And the reason for this is that this type of panel that are molecular profiling panels do not work well with hepatotoxicity targets. And the reason is simply, one, there is no one single target that causes hepatotoxicity. So now what I want to do is I want to show you some examples on how to use molecular profiling to identify adverse events that will be that can affect the outcome of a compound. The first example I'm going to show you is a drug called amoxapine. And amoxapine is a tetracyclic antidepressant of the dibenzosazepine family. It's a secondary amine antidepressant, and it actually is currently on the market. The main target for amoxapine is serotonin transporter, which is going to be this one, and the norepinephrine transporter, like many other antidepressants, this one, right? But unfortunately, if you look at this graph, by the way, what you have here is this is a typical pharmacological profiling. In the x-axis, you have every target that was tested in this pharmacological profile. In the y-axis, you have the percentage of control. And one of the things that you can see is that this compound is not very clean, actually. You have a lot of activities that are being hit by this target and by this compound, and that is a very, I would say, very complex pharmacology. So one of the targets that are being hit, and you can see here with the red arrows, is the dopamine D1 and dopamine D2. And unfortunately for amoxapine, in this case, one of the other events that you see whenever you have antagonism in the dopamine D1 and dopamine D2 is the possibility to have this syndrome. It's called the neuroleptic malignant syndrome. And the problem here is that it's a very severe stress syndrome in which you have you have autonomic instability. Basically, you lose control of all of the autonomic systems, and that can lead to death. And unfortunately for amoxapine, that is one of the adverse events of this drug. And they have seen in the clinic that in several cases, actually, that's something that is observed. So the next example that I'm going to use is a drug called memantine. And memantine is a novel class of Alzheimer's disease medication, and it blocks the NMDA type glutamate receptors. And here what you can see again is the same type of pharmacological profiling. These are the targets. This is the percentage of control. And what you can see is that this is a very clean drug. Only targets the glutamate NMDA receptor. Now, you could say that this is a good drug then, right? But unfortunately, several years ago, about 15 years ago, a program led by scientists at Merck, they developed this drug called MK-801, and MK-801 was developed actually blocking the glutamate NMDA receptor. The program never made it to the clinic because when it was tested in rats, these animals developed backwards in the brain, and they call it the only type lesions. And this was enough to derail the program. Again, unfortunately for memantine, this is exactly what they have seen with memantine, and this was reported to the FDA last year. And you can see that just by doing the pharmacological profiling, you would have been able to predict this type of activity. The next example that I'm going to show you is a drug called BW723C86. And this is an anxiolytic drug. It's an investigational drug, and the target for this drug is going to be the serotonin. It's going to be serotonin 5-HT1A and 5-HT2. And that actually is shown in this pharmacological profile. You can see that the drug actually does not have a very dirty profile. It only hits a very few set of targets, but you can see the activity right here on the right side of your screen. This is 5-HT1A, and this is going to be 5-HT2B. In this case, the issue with this drug is going to be that you're hitting the serotonin 5-HT2B, and the problem here is that if you have an agonist, if you have an antagonist of the serotonin 5-HT2B, your company is going to be perfectly fine. But if you have an agonist of the serotonin 5-HT2B, then you have a risk of valvulopathy. So how do you determine that you have an agonist and not an antagonist of a severe liability like the serotonin 5-HT2B? So let me show you how to do that. By the way, in this particular case, I'm going to tell you the answer. This is an agonist, and you will see the liability of valvulopathy for this receptor. But this is how do you go about determining if your compound is an agonist and antagonist. And this is going to explain now why it's so important to have a very broad pharmacological profile. That is the reason why we have 1,200 or 1,500 of this profile. It is because we want to provide you enough assays to build confidence that you have enough information to make the decisions. The first step is, after you run your pharmacological profile, you obtain information on the primary screen. Remember, most of the time this is going to be done at one concentration. In this case, for this particular drug, you have 100% inhibition in the serotonin 5-HT2B. But the next question is it potent? You don't know if the compound is going to be potent or not, and that is why it's so important to do an IC50. In this case, we can see that the activity of this drug in the serotonin 5-HT2B assay is very potent. But since now you have a compound that inhibits the serotonin 5-HT2B and is very potent, now it's very important now to make the determination: is my compound an agonist, or is my compound an antagonist? If it's an antagonist, I'm fine. If it's an agonist, now I have the risk of developing valvulopathy. So now we have this assay. It's called the serotonin 5-HT2B IP, and IP stands for inositol phosphate. In this assay, we measure the activity of the compound by following the inositol phosphate pathway. And what you can see, the outcome of this assay for this compound is that it shows that the compound is actually an agonist, and the EC50 of this compound is very potent, is 66.4 nanomolar. But now the question is this enough information for you to feel confident that the compound should be taken off the drug discovery pathway? Well, in order to build confidence, we have another assay, and And this is a very important assay because the reason why you develop a blood assay, you have the cardiotoxicity caused by the serotonin 5-HT2B is because you have proliferation of cardiomyoblasts in the heart, and they're going to migrate to the valve, and then you're going to develop the cardiotoxicity. So this assay now measures the cell proliferation of cells that contain the serotonin 5-HT2B receptor. And what you can see that the outcome of this assay is that the compound BW723C86 is actually able to induce proliferation. So now you have two pieces of information that are very telling that this compound will indeed cause the activity. By the way, this compound never made it to the clinic. And we can say that we have the same type of testing paradigm for every one of the assays in our portfolio, primary assays in our portfolio. So, and I'm going to give you just one example, very quick example, that we can offer the same type of decision tree for the hair potassium channel, another very important target. And in this particular case, here, what I'm going to go very quickly on this, is that we have the primary screen, which is going to be a binding assay for the hair potassium channel. If you have a hit in the primary screen, then you're going to do an EC50 in the same assay. If your compound is potent and warrants that you do a follow-up on this target, then we have an assay that is going to be a functional assay. This is going to be an electrophysiology assay. This is the automated patch clamp assay, in which we can tell if the compound is able or not to block the hair potassium channel. But the question is this enough information, again, to take a compound out of the pipeline? And the problem here is that not all the compounds that block the hair potassium channel will cause arrhythmia. The problem here is elongation of the QT signal, and that in turn is going to cause arrhythmia. So in order to be confident, we have another assay, and this assay is done using human stem cell cardiomyocytes that are tested in a multiple electrode array. And what we measure here, we measure two things. One of the things that we can measure is beating, and you can see actually the beating of these cells. And then we can determine, does my compound affect the beating? But this is ultimate what you really want to measure. And you can also measure the contribution of all the channels in the heart by measuring the total current in the cell. The reason why this is important is because drugs, for example, like verapamil, they can block the hair potassium channel, but they do not show the QT syndrome. They do not cause arrhythmia. And the reason is because you have compensation by other channels in the cardiomyocyte, like sodium and calcium channels. So we can do that. We can measure the total effect of this compound in these cells. So with this set of decision tree now, we can give you enough information to make a more informed decision regarding your compound. But what else can we do? So since we want, what I showed you before is that by testing by carrying on a pharmacological profile, you build a lot of information about your compound. There is something else that we can help that can help, and this is utilize the use of pharmacoinformatics to try to answer this question. Is there anything? Is there more information that I can have about my compound by the use of these tools? And these are the basis for the pharmacoinformatics approach for the use of molecular profiling information. Imagine that you have a test compound that you run through one of the pharmacological profiles, and now you imagine that you create a signature of this pharmacological profile. And imagine it is going to be your compound or it's going to be a test compound. Now, we have databases, we and others. Our database is called Foresight, but others can be called Bioprint, and there's several in the industry. In our case, we have a database composed of 1,500 compounds, and each one of these compounds was tested in the same pharmacological profile as your compound. And now we can create a signature of these 1,500 compounds that are in the pharmacological database. Now we can compare it to the signature of your test compound. By doing that, if two signatures are similar, now we can say that perhaps the activities are going to be similar, and now we can predict an in vivo effect. But if two signatures are different, two pharmacological profiles are going to be completely dissimilar, then we can say that the in vivo effects are going to be different. So that is the basis for the pharmacoinformatics approach for the analysis of pharmacological profiles. Our pharmacoinformatics tools is called Foresight, and it's been available since 2010. And what it does is predict in vivo effect from in vitro pharmacological data, just like the one that I showed you before for SafetyScreen or for any of the panels. The Foresight components are four. First one is the database, and the database is composed of 1,500 biologically active compounds. Other databases that are on the market are Bioprint, for example, from CERREP, and that is composed of just a bit more, maybe 2,000, I think about 2,000 biological active compounds. Every single one of these 1,500 compounds was tested in pharmacological profiles. In this case, was lead profiling screen, and we have annotations for each one of the compounds. The more important component of the database is the web interface, because it allows the user to interact with the database to gather the information that is going to be used for the prediction of in vivo activities. But how does this work? And I'm going to show you with one example, and this is going to be in the next slide. So I'm going to show you two examples. I'm going to call them test 1, and the other one is going to be test 2. And the first example is test 1. You can see that it's a typical pharmacological profile. It's just like the one that I showed you before. And you can see that this particular compound hits two sets of targets. One is the adrenergic receptors, and the other one is going to be the dopamine receptors. But just based on this pharmacological profile, can you determine what the compound does and what are the adverse events? No, really cannot really tell. All what you can say that you have dopamine and adrenergic activities. But now, if we take this pharmacological profile, we create a signature of the profile, we compare it to the signature of every compound in the database, in Foresight database, remember 1,500 of them, and in order to do this activity, we utilize an algorithm. In this case, we're going to use the cosine correlation. And what the cosine correlation is going to do is going to compare these signatures, and it's going to give you a number. And the number is going to go from -1 for completely dissimilar to 1 for identical compounds. So if two compounds are identical, the number is going to be 1. Two compounds are different, they're going to be -1. And any number above 0.8 is going to be very meaningful. So what happens if we do that? Well, you can see that compound test 1, when compared to every compound in the database, was able to identify several compounds with a value of 0.8, but the more highly similar compound is going to be pyramperon. And you can see that the correlation coefficient for this in this test is 0.924. This is very, very similar. And what pyramperon is an antipsychotic acting in the dopamine T2 receptor, and this is what you see. So for pyramperon, one more piece of information is that the adverse events are mostly hypotensive, extrapyramidal, and sedative. And now and those are exactly the same activities that you're going to see with compound test 1. Compound test 1 is risperidone, and the risperidone is also an antipsychotic drug, and the other events that you see in the clinic are in the same rank order, which is hypotensive, extrapyramidal, and sedative. So here you have an example in which you can take a pharmacological profile, and just utilizing that information, you can determine the activity of that you're going to see in the clinic of this compound, and you will also see the adverse events that are going to be seen in the clinic. So now let me go to compound test 2. So compound test 2 now is a smaller profile, and here is the nice thing about bioinformatics. Bioinformatics tools tend to be very flexible, and you not always have to screen in exactly the same panel as the panel that the database was tested at. Of course, the more compound the more target that share between the database and the testing panel, the more resolution and the more meaningful data that you're going to get. In this particular case, you have a 40 target panel, and you can see that the activities that you see with this compound test number 2 are mostly in the calcium channels here and histamine receptors. Again, it's very difficult to make a determination on what this compound really is, just based on the pharmacological profile. But now, if we were to take this compound and create a fingerprint again, this particular compound, and compare it to the fingerprint of every compound in the Foresight database, utilizing the same correlation as we used in the previous example, which is going to be the cosine correlation, the results are going to be shown here. It's at the highest similarity, remember the 0.8 value, the highest similarity that we see between compound test 2 and the database is the compound GBR 12909 with a value of 0.8. So again, this is very meaningful. And what this compound is? So if we go and look at it, compound GBR 12909 is a piperazine derivative, it's a dopamine reactive inhibitor, and the important thing in this particular example is that the compound was halted in phase 2 clinical trials because of the long QT syndrome. Interestingly, what the compound test 2 is actually terfenadine. Terfenadine is an antihistaminic, and that's why you see the histamine H1 activity. And more interestingly is that the compound was withdrawn from the market because of long QT syndrome. So here you have an example where if we would have done the same type of analysis early on, the market would have never made it to the clinic and would not have to be withdrawn from the market. So in summary, what I've shown you is that is the reason why in vitro pharmacological profiling is so important. And the reason, again, just to reiterate it, is because allow for the early detection of clinical liabilities. And in vitro pharmacological profiling is very cost effective, and this allows for more compounds to be tested, including metabolites. And panels can be designed for specific projects and different stages of development of each compound. And very importantly, bioinformatics tools can be used to predict in vivo activities. Thank you.
Great. Well, thank you, Dr. Castillo. That was a very informative presentation. I would now like to invite our audience to continue sending their questions and comments in right now using the questions window for this, which is the Q&A portion of the webinar. And Dr. Castillo, I've already received some questions, so I'm going to start with those. Our first question, can I use a SafetyScreen to predict off-target effect of biologics, including antibodies?
Yes, and actually that is a very important question because biologics are actually able to they are able to interact with targets. One of the things about biologic is that because of the way that enter the body, they are immediately exposed to many targets in the human body. Basically, you don't have to penetrate barriers. So many targets actually will be able to interact with these biologics, and we see that in many profiling examples. And in particularly, the targets that are important for these activities are going to be vector receptors. Also, very important targets are going to be in the immune system. And more importantly is that you not only have to limit to panels like the one SafetyScreen, but you also have to increase the scope of the testing to include immunotoxicities.
Okay, thank you. Our next question here. Audience member says here, I am working on a GPCR target. Do you think it is necessary to test in a SafetyScreen which includes GPCR and enzymes, or just test in a GPCR panel? Your thoughts on that?
Yes, so actually it is very important to test in panels like SafetyScreen that include enzymes and other ion channels, and in spite of the fact that your target is GPCR. And the reason is simple. The reason is because the off-target activities most of the time are not related to the target that you're testing on. The off-target activities will come from targets that are non-related to your target of interest.
Right. Okay. Thank you. Our next question here: how was the SafetyScreen validated in terms of its ability to predict clinical effects?
The SafetyScreen was validated by looking at every single one of the type A adverse events that have been shown to be able to really program, and this was done actually using literature. So we scope every single one of the targets that is a reason why a compound has been taken out of the guideline. We looked into compounds that were withdrawn. We looked at compounds that were taken in phase 1, phase 2, phase 3, and we also look at compounds that were taken early in preclinical discovery.
Right. Okay. Thank you. Our next question: does Panlabs offer in vivo CV testing to estimate safety margins when a positive result is seen in the in vitro HERG test?
Yes, we do actually, and that is one of the very important panels that we do offer in our facility in Taiwan, and can really help to complement the testing that we do for our SafetyScreen.
Okay, thank you. Our next question here: typically, what concentration, for example, 100 nanomolars, should be taken for the first I believe that's HTS screen of 87 proteins in your panel? I can repeat that if necessary.
The answer actually for that question is as high as you can, and the reason is because normally it's going to be around 10 micromolar. Above that, actually, you're going to run into issues like solubility. So 10 micromolar is what we recommend, but the reason why it's important to test at very high concentration, whether it's only lower than that, is because we do want to make sure that if you have an activity, you see it, because then if you see it, then you can determine, for example, the potency, you can determine safety. But if you miss it, then you may run into trouble later on let's say, later in drug discovery.
Right. Okay. Thank you. Our next question here: why use binding assays instead of agonist or antagonist determinations as the initial screen? Your thoughts there?
Yes, there's many advantages of using binding assays. The first one is binding assays allow you for the determination of affinities, and this is important because you can use that information to use that information for SAR, and it's very difficult to use that type of information from let's say a cell-based assay in which you're going to obtain agonist and antagonist information. The second reason is very cost-effective. So in one experiment, you will get the information of the interaction of the ligand of your compound to one particular ligand. If you were to do the same in a cell-based assay for agonism and antagonism, you have to do at least double the effort. So they tend to be binding are really more cost-effective. The other reason is because binding assays also are a direct measure of interaction. And in a cell-based assay, what you're measuring is a reported type of activity, and you may miss some compounds. So those are the reasons why it's better to profile using binding assays.
Right. Okay, thank you.
Just one more thing. In other words, is the closer that you are to the target, the better that your prediction is going to be.
Okay, great, thank you. Our next question here, can you elaborate on how you determine the safety margin for HERG?
Safety margin? So that is a good question, and I'm going to try to give you my best answer. And the reason for this is because we don't have, and I just want to make sure that I'm understanding the question correctly. So the question is, so the question that I'm going to answer is let's say you have your potency and you have a hit, right? What is going to be you have an adverse event related to one target, and you have the affinity of the target of interest. What is the safety margin? So in general, 100-fold difference will give you enough of safety margin. Never go below 10, and 1,000 would be even better, right? So 100-fold, I would say that is the minimum that you have for this type of margin.
Okay, thank you. Our next question, can you give more detail on the in vivo CV test? Just maybe elaborate a little more on that.
I'm sorry, could you?
Yeah, absolutely. I'll say that again. Can you give more detail on the in vivo CV test?
Actually, I'm going to have to refer this question to my colleague Jamie Van Gardner. So he's actually one of the experts in vivo. I'm really more focused on molecular pharmacology, so he's going to be able to give you a better answer on this.
Sure.
Yeah. So essentially, what we currently have in our portfolio are a few rodent tests. One would be the guinea pig QTC. That would be the direct correlation to the hurt questions that have come up, and that's an assay that is very predictive and correlative to the development assays, as well as what you would see in the clinic for testing for QT prolongation. We also have a battery of rat telemetry tests that will measure other effects on heart rate, blood pressure, etc., as well as a respiratory battery. So essentially the safety pharmacology assays you'll be taking a look at in development can be performed inexpensively in a non-GLP fashion in rodents. The last piece as well that Gonzalo mentioned was actually taking a look at cardiomyocytes that we are in the final stages of validating that would actually be a cell-based assay that would predict those cardiovascular events. Now, if this answer is not sufficient, what I would encourage the person who asked the questions to contact us, and we would actually have a specialist with respect to safety pharmacology help design an experiment or help answer questions in a much deeper fashion.
Okay. Well, thanks for that, Jamie. Our next question here. Are all the receptors, enzymes, and transporters of human origin?
Not at this time, actually. But however, we're working very hard in trying to move into that situation. So our goal is to have every single one of the enzymes, receptors, and transporters of human origin. In some cases, unfortunately, we do not have, for example, glutamine receptors and GABA. We do not have human receptors, but we're working with that, and our goal is to have by next year all of our targets human origin.
Right. Okay. Thank you. Our next question: what's the typical turnaround time after receipt of a compound on your end?
10 days. So normally, actually, for this particular target panel, SafetyScreen, the turnaround time is 10 days, 2 weeks.
Okay. Thank you. Our next question here: how do you relate the data with an in vivo effect if we have a compound that's having a nanomolar affinity in binding, but in functional reporter gene assay of a micromolar affinity? So, sorry, I was kind of paraphrasing there. I can repeat that if you'd like.
Yeah, could you repeat that question?
Yeah. How do you relate the data with an in vivo effect if we have a compound that's having a nanomolar affinity in binding, but in a functional reporter gene assay with a micromolar affinity?
So many questions actually. The first one is in a binding assay, you're measuring the direct effect, the affinity of the compound to the receptor. So in a cell-based assay, it's a reporter type system, and often there is other. So it's going to depend on the reporter system. For example, is it going to be a calcium flux or something like that? So there's many other components that can affect the outcome of this result. For example, are you measuring the how you detecting the effect on the membrane, or does the compound have to penetrate the cell in order to see the activity? For example, for nuclear hormone receptors. So there's many more. It's not a direct measure of affinity when you go into a cell-based system, as opposed to in a binding assay where all what you have is a receptor and compound. In other words, it's a very elegant system compared to a cell-based system.
Right. Okay. Well, thank you very much. We have reached the end of our question and answer period. If you do have any further questions, you can direct those to the email address showing on your screen for Dr. Gonzalo Castillo, and that's Gonzalo Castillo at Eurofins.com. Thank you, everyone, for participating in today's conference. You'll be receiving a follow-up email from XTalks with access to the recorded archive for this event. A survey window will be popping up on your screen, and your participation is appreciated as it will help us improve our further webinars. Now, please join us in thanking our speaker, Dr. Gonzalo Castillo. Dr. Castillo, do you have any closing remarks?
No, I just want to thank you. Thank you very much for participating in today's webinar. And I appreciate your questions. So if you have any other questions, please feel free to contact me. You can have my email, and you can also reach me at the Eurofins Panlabs directly. Thank you very much.
Great, thank you. We hope that you found this conference informative. Have a great day, everyone.